Hi dear student, welcome back to another informative lecture of biological molecules. The SLOs we will discuss here are about carbohydrates. Carbohydrates are organic molecules composed of carbon, C, hydrogen, H, and oxygen, O, in a ratio of 1:2 to 1. They are one of the four main biological macromolecules, alongside proteins, lipids, and nucleic acids. They are energy source for living organisms. In next SLO, let's classify carbohydrates. Four major classes of carbs are monosaccharides, disaccharides, oligosaccharides and polysaccharides. Monosaccharides, simple sugars, for example, glucose, fructose and galactose. Glucose is the main energy source, also known as blood sugar. Fructose is the sweetest sugar, found in fruits. Galactose, which is found in milk and later on converted into glucose. All these monosaccharides are important for living organisms. Glucose and galactose have aldehyde group, CHO. Fructose has ketone group, C=O. Disaccharides are formed by two monomers or units by a glycosidic bond through condensation reaction. Examples. Maltose is formed when two glucose molecules combine together. Sucrose, glucose plus fructose. Lactose, glucose plus galactose. Oligosaccharides are carbohydrates composed of a short chain of 2 to 10 monosaccharides, simple sugars, linked together. They are a type of saccharide polymer, found in various foods, and can function as prebiotics in the gut. Polysaccharides are major classes of biomolecules. They are long chains of carbohydrate molecules, composed of several smaller monosaccharides. These complex biomacromolecules functions as an important source of energy in animal cell and form a structural component of a plant cell. Let's summarize in a fun way. Monosaccharides are the tiny toddlers, single sugar units. Glucose, fructose, galactose. Simple. Disaccharides are like two toddlers holding hands. Sucrose, table sugar, lactose, milk sugar. They form when two monosaccharides do a dehydration reaction. They link up and kick out a water molecule. Oligosaccharides are the middle kids, 3 to 10 sugars. These are the sneaky ones in beans that make you gassy because we can't digest them. Polysaccharides are the giants, long chains. Starch, in potatoes, glycogen, in your liver, cellulose, in plants. Some store energy, some are like biological Lego bricks. Next up in our discussion is explanation of monosaccharides. Let's distinguish L and D glucose. D and L forms of glucose, which are stereoisomers, are distinguished by the spatial arrangement of their hydroxyl, O, groups, specifically at the chiral carbon farthest from the carbonyl group in their Fischer projection. In D-glucose, the O group on this chiral carbon is on the right side of the projection, while in L-glucose, it's on the left side. A chiral carbon is one that is attached to four different groups, making it a stereogenic center. In glucose, the chiral carbons are the second, third, fourth, and fifth carbons. D-glucose is the natural one, your body uses it. L-glucose is its mirror twin. We can make it in labs, but it's useless biologically but can be used as artificial sweetener by diabetic patients. SLO 1.4.5 demands to illustrate different structures or forms of D-glucose. When glucose forms a ring, carbon 1 becomes a new chiral center, the anomeric carbon. Alpha and beta glucose are two isomers of glucose, differing only in the orientation of the hydroxyl group at the anomeric carbon, carbon 1. In alpha glucose, the hydroxyl group points down, while in beta glucose, it points up. This difference in orientation affects their reactivity and stability, with beta-glucose being more stable and less reactive than alpha-glucose. OH downwards makes alpha-glucose. OH upwards makes beta-glucose. These tiny flips can change everything. Starch uses alpha-glucose and it is easy to digest, while cellulose uses beta-glucose and it is indigestible. That's all, my dear student. Stay tuned for more. Don't forget to subscribe.